Recording in progress.
We have set previously at 3:45. So please, ladies and gentlemen. Attention please, ladies and gentlemen, for those who are standing outside still, please rejoin and come to our room since we'd like to hold the next session, our last session, which is the Symposia 5. So please come and join us since we would like to start at 3:55. No, 3:50. Mhm.
Okay, good afternoon, everyone. Welcome back from the coffee break. We are now entering the second part of Symposium 5, which is the presentation from invited speakers and, yes, from invited speakers. But before we give the floor to the chairperson, we would like to take a moment to acknowledge and express our gratitude to the organizations and sponsors that have made this event possible. Let us recognize
The first one, we are organized by the Faculty of Forestry, Gadjah Mada University.
the Ministry of Environment and Forestry,
And we also have a number sponsors, which are April Group,
Global Environment Facility in partnership with United Nations Development Program and Combating Illegal Wildlife Trade,
Peat and Mangrove Restoration Agency of the Republic of Indonesia,
Borneo Nature Foundation,
Taproba Nika, the Journal of M.Sc and Biodiversity,
Flora and Fauna International,
Copenhagen Zoo,
Kaleka,
Action Conservation Foundation,
and Pesona Indonesia Tours.
and we are move on to the next session, which is the Symposia 5, with the grand theme, Innovation in Biodiversity Conservation Technology. And we have two keynote speakers. Will mention by our chairperson there, again with Dr. Francisca. And okay, without any further ado, while we are waiting for the other participants who will be joining here, I give this Symposia 5 to Dr. Francisca directly. Thank you.
Thank you to MC. Welcome everyone. I think we are still missing our second keynote speaker, Professor Hong from Korea. Let me just check. Prof. Lilik, sebentar ya Prof. Bisa ditelepon? Oh, coba tak kontaknya. Saya telepon Pak.
All right, ladies and gentlemen, because we are still waiting for the speakers, we would like to wait around 3 minutes to wait the speaker come. All right, then. And while we are waiting as well for the participants come in, and for the organizing committee, please let they know about our discussion here in Symposia 5, that the room Arjuna 1 is starting now. To the organizing committee or liaison officers, for the speakers, please to reach out and also follow up for our speakers as well. The one keynote speakers and one invited speaker here, and actually the one here is attended, right? And we would like to look for the rest one.
Okay, thank you. Good afternoon, ladies and gentlemen. We'll have our last symposia of the day, Symposia 5, entitled Innovation in Biodiversity Conservation and Technology. This afternoon, we will have three distinguished speakers from their own expertise. First up, I would like to kindly invite Professor Lilik to join me on the stage here. Professor Lilik Budi Prasetyo is a professor in landscape ecology and head of environmental analysis and geospatial modeling at Department of Forestry Resources, Conservation and Ecotourism at IPB University. Please welcome Prof. Lilik. He is also the coordinator of the working group of the Lapan IPB satellite, Lapan A3. His principal research interest is in the application of remote sensing and geographical information systems for forest conservation. His research is often related to landscape ecology, especially species distribution model, biodiversity indicators, land use and land cover changes, forest monitoring, carbon sequestration, and REDD+. His work has been presented in seminars, workshops, and published in several national and international journals. Without further ado, may I kindly ask Prof. Lilik to give his talk this evening.
Terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for your invitation to share the experience of related to the innovation of wildlife conservation. Please. Yeah, there are numerous technique or innovation that already published in the papers, so many, and I selected at least three innovations related to the monitoring of species and habitat, and then relate to the data analysis and processing, and the third is about the community science. Troubles? Okay. Yeah, the first is regarding the monitoring and monitoring of species and habitat. Please proceed, please. Next. About the monitoring of species habitat, we concerned about the application of remote sensing. And when we talk about remote sensing, we actually talk about the platform and sensors. So basically the platform of remote sensing, based on their characteristics and the positions, can be divided into three: ground-based remote sensing, airborne-based, and then a satellite-based. Yeah, for example, ground-based, we know about the spectroradiometers, and then in airborne-based, we know the platform is like a UAV, and balloons, and also flights. And then in satellite-based, we can see a numerous type of satellite. And if we see the sensor, we can identify that some sensor is supposed to be related to the mission of the satellite. Yeah, basically there are RGBs, meaning that the sensor can capture from 400 to 700 nanometers, and also there is a radar, LiDAR, and also thermal. Yeah, this is depend on the mission of the satellite. Still. Okay. Actually, when we talked about the first is about the regarding the platform and sensor. If we talk about the airbornes, we see a lot of innovation there. Okay, yeah, this is what I explained just Yeah, we can see the definition of remote sensing and so on. Next, please. Yeah, when we talk about the base remote sensing based on satellites, this is already in operational. So many kind of satellites and also many sensors that have temporary resolution is diverse from 16 days, 5 days, 1 days, also to times a day is depend on the missions. So some of them is free. Oh, sorry. Free, you can download it from the Earth Explorers. So what remote sensing can be used? First is about the habitat change. So we can identify, just because satellite remote sensing captures regularly, so we can follow the changes of habitat, and also we can, for example, measure the fragmentations, deforestations, and others. Also, the image can be used for other environmental variables in the modeling. For example, it's normalized different water index, land cover, normalized Purna index, and so on. And also can be processed to be canopy cover. And how about the airborne-based remote sensing? First, it's about the first sensor is RGB. The platform of airborne can be in the form of quadcopter, fixed wing with vertical and horizontal takeoff, and also fixed wing with belly landing. So we can set up with another acquisitions or of another acquisitions. So if we set up this, so we can use, for example, the characteristic of lacking tree of bird paradise, for example, we can construct the point clouds based on this, and using SVM, we can then model the surrounding area of these trees. And can the RGB sensor can see wildlife? Yes, possible, but if under canopy, no chance. And if we take a look more closer, we can more getting clear objects. And for example, in this case, for example, we need kind of object segmentations to detect where is the species and what kind of species there. And for example, orangutan, somehow we can detect if they are stay in the canopy cover, top of canopy cover, bekantan also can be, elephant, yes. And how about the thermal sensor? Yeah, we tried to do this with our students, Ahmad Abdul Aziz. We try in Halimun National Park. This is using the manual flight. The distance about 35 to 50 meters. And we can see by RGB sensor, we can see Lutung, Owa Jawa, and Surili. Yeah, with very clear. So for this, for example, and this. But in terms of thermal area, is still problems, just because the resolution is not good enough. Just because usually the resolution of our ground spatial distance is about 20 to 30 centimeters. So it's impossible to do segmentation of based on the object shape, yeah. So if we do like this, so the based on the experience of this site, so somehow the wildlife is running away just because they are feel to the disturbance. And how about the nadir acquisitions? The same still. especially if RGB, we can still detect the Owacho and Sururi, but for the thermal sensors, we have difficulties, especially in the species that are living in under the canopy. So, but if we conduct observations using nadir, we can see that the wildlife doesn't feel disturbed, so it's better. How about in the savanna? We have a chance to test this kind of flight, a drone, fixed wing. Trinity is about maybe 6 bands there, panchromatic, multispectral, and thermal. Yeah, multispectral we have red, green, blue, and near infrared. So we have about 6 to 7 bands, and we fly over the Pulau Rinca. And what is the result? Like this. So we can detect using RGB, for example, buffalo, deer, and komodo. But for the thermal, it's a bit difficult. It's impossible. So because the komodo body temperature is somehow lower than the environments. Yeah. And this is not a good time just because at the same time, our friends from the KSP, Komodo Survival Program, doing the same thing. They capture using a camera trap. So most of Komodo goes to their traps and no one in the savannah area. So we can't see them. Only once, this very near, two in this spot, this spot, yeah. And this is LiDAR. So we have opportunity to test this kind of drone occupied, mounted with LiDARs. So using the algorithm developed by IMA, using this kind of algorithm, then we can construct the point clouds, and then with point clouds, we resize into pixel size, yet the same with Landsat, and then we make correlations. And from sample points, then we can extrapolate to the wider area. So we got this. This is comparison between canopy cover developed by Hansers, Maryland University, and our canopy cover. And this is the whole analysis that we conducted last time. We already published in this web. So we check. You can check these products. So the important thing of this is should be check the accuracy of the estimations. So far we only test the accuracy in Sumatra and West Java, not the whole area. This radio telemetry, yes, this is based on radio. Our students test in Rawaupar National Park. They mounted here, and so they bring this antenna to detect the position of the kura-kura Ambon. So we got this. So we can detect any movements, daily ring, dispersion, and also habitat information, just because this device can send also the temperature of surrounding area. Okay. How about the Argo system? Argo system. Actually, Argos is a system that can be divided into some component system, which is a transmitter that mounted in the birds or other objects, satellite, which is NOAA and EDOP of France, and then receiving station, ground receiving station, and data processing unit. So every day, the device transmitted the informations captured by satellite, and then sent to the ground and processed by ground station and sent to the scientists. So this is the flow of Hachikuma or Oriental honeybirds buzzards or Pernis petilorin kust. Yeah, setiap saat, every time they visit us, especially when the Japanese in cold winter or winter and cold seasons. So using this data, so our students try to make a suitability habitat for breeding season and wintering seasons. And using the IPCC scenario, SSP2 and SSP5, so we can predict the shift habitat due to the global climate changes. So in detail, you can read in the global ecologies and conservation papers. And data processing is related to cloud computing. This is one example. For example, this is a Maxent. And this is Condatis. This is developed by Liverpool University. Johnny Hudson's. They provide tools for decision support tools to identify the best location for habitat creation and restorations to enhance existing habitat networks and increase connectivity across landscape. So basically, first we have to develop habitat suitable model, and then we can detect the source and the sink of species, and we can running the model. So we can see the barrier, so that barrier should be improved. And when we improve, so we can see the flow of movement, generation movement actually, not the short time, but generations. We can see the movement and more smoothly than before. So they move just because disturbance. In this case, we assume that there is a global climate change, and they should move to the previous habitat suitable to the other place, and we can follow the flow, and we can see also the barrier during their movement. Data mining is also innovation that can be used. Just because this is what happened now. In 1 minute, there are so many data flows in our internet. This is a statement by some researchers in the 1980, I think, and now it's still relevant. So this is the fact that so many mobile phones, so many process in the web, and this is like a tsunami of data. Yeah? So, and how we can use this for conservations? Sorry. Nowadays, we know the term data mining, or we can see knowledge discovery in database. This is in the definitions, and this is an application mostly for financial banking, healthcare, transportation, marketing, and environment conservation. And this is the papers using data mining for identify the conflict places of harimau and people. So this is using R, R. And the third innovation is community science, as in the morning session already explained by Burungnesia, Burungnesia. This is a similar one. This is an e-naturalist. So everyone can contribute to the position or operation of species, and they can upload the photos, also the position already this 156,000 observation data, many. So this is one of our students use this data to to map the suitability habitat of katak tanduk ini. This is for katak tanduk, and this is the result. This is the habitat that very high suitable, and the other is not. Yeah. Okay, for the summary, development of remote sensing platform and sensor open up opportunities to monitor habitat and species more quickly and accurately. And the digital technology innovations trigger the development of enormous data on the internet. Despite the negative impact of this phenomenon, huge data can be processed into useful informations for biodiversity conservations by utilizing cloud computing and data mining. People can play a role in species conservations with community science innovation by utilizing mobile or smartphones. I think it's all. Thank you very much for your attention.
Thank you very much for the inspirational talk by Prof. Lilik. He presented a very broad topic actually about innovation in biodiversity, and he presented from the basic understanding that there are three items on technology innovation. Firstly, about monitoring of species and habitat, and then data processing, and lastly, community science. He also mentioned a lot about how current techniques on monitoring of species and habitat. I'm personally familiar with radio transmitter for orangutan, so I have questions for you. And of course, there are more advanced than radio transmitter technology, and now people are using satellite-based. And then the data processing is also growing quite advanced recently. And lastly, discussion about community science that's already been touched on this symposium for a couple of times. A few examples, Purna that are using data community science quite extensively, and Prof. Lilik also mentioned iNaturalis. And all these innovation in technology help us to better understand about the ecology of wildlife and the ecosystem, about movement, migration, habitat quality, and all that can be synthesized into more practical improvement in management of both wildlife biodiversity and our human management. For example, because I come from a health background, I'm always thinking about One Health and how this information about wildlife, their diversity, their population, they're always connected with human. And the concept of One Health means that one can only be healthy, human can only be healthy if the wildlife and the ecosystem is also healthy. And we need a lot of data to prove that and to improve it if there's room for improvement. So I'm gonna open up time for discussion. Anybody would like to give comments or question about innovations and technology on biodiversity? Silahkan.
Yeah, sorry, Prof. Lilik, thank you for the talk. So I have a question. Like I used to study in Oxford, and I have like a lot of colleagues in India, Africa, and South America, and a lot of research in conservation. They use like a lot of like new technology already. In Indonesia itself, we don't really use that technology yet. For example, GPS collar has been used in Africa for a long time, but in Indonesia itself, like using GPS collar for tiger, for example, is very hard to do. I'm just wondering like why there's like a kind of like a slow progress in using like newly developed technology in wildlife conservation. Is it because of the funding, the government regulation, or else? Thank you.
And your comment also, I echo your comment because as I said, I work with orangutans and we can only use radio transmitter because satellite is expensive and I don't know, Prof. Lilik, please, enlighten us.
Yeah, as far as there are GPS collar already implemented in Indonesia, for example, and when the releasing the capture harimau Sumatera from Aceh to Bukit Barisan Selatan, they also use GPS collar, and also in Riau, if we see the data of BKSDA, we can find some individuals of elephants already equipped with GPS collars, and also some Sumatra triangle in Bukit 30, sorry, in Kerinci, yeah, and also in Padang, they are also GPS collar. Yes, the barrier is about the price, yeah, the price, and also I don't see the barrier of implementation of the technology, just because if you do this, and it would help the BKSDA to monitor the home ring and others, yeah, and also to can be detect the conflict area, and also set up the corridor, etc. So many useful output from this kind of activities and research. There is no any barrier, but I think cost, yeah, cost. For example, for Argos systems, one transmitters is about $5,000. Yeah.
Dan harus
And-
langganan ya, Prof ya. untuk
Yeah, and
belajar.
you have to spend $5,000 for, I think, 1 year, yeah. 1 year, that's the cost.
Sure. Yeah.
Yeah, there's one of my friends like who's doing the GPS collar for tigers, and she kind of like told me like one of the issue is like because we're using a provider from outside, from Europe, there's like a concern the data is not collected directly to the government, and the government haven't consideration a concern about the safety of the data, because the data from the satellite needs to go to the provider in Europe and then goes back to us in Indonesia. I'm just wondering like if there's also like a consideration that is happening at the moment, or
Yeah, so far I don't think it's happened, yeah.
Yeah.
Just because not everybody is try to put the GPS collar and so many individual, yeah. I think it's no any. But sure, the data should be goes to the governments,
yeah,
not the NGOs and or others, yeah. Should be goes to governments, and government gives the data. So if we want to use this data, so we have to apply with official later.
yeah.
Yeah.
So maybe like a better flow for the data collection and transfers
Yeah.
needed.
Yeah.
In the future. Yep.
Just because somehow it's very crucial, yeah, for poaching and hunting. Yeah.
Thank you.
Thank you. From the current discussion, we identified that some of the main challenges are about the cost of the funding, and also about the data sharing. As we know that data sharing can be good if you share good data, but that can also be misused by not good intentions. And we also touch a bit about the government's role in the use of technology. And in the online participants, I have one participant from Taman Nasional Merbabu, who is from government. Can I please kindly ask Mr. Nurpana to open his audio and
Okay.
share with us?
Okay, thank you for the opportunity. This is a very, very good topic, I think, the innovation about the wildlife monitoring system. And I think the problem not only about the cost, I think, but also how to manage, how to process, how to analyze the data. And I think that's very important because the problem maybe in my institution, maybe we have installed camera trap systematic, yes, and we have so many data, but we have a challenge how to analyze, how to process the data. I think that is the problem, I think. How about this problem, Prof. Lilik? Thank you.
Thank you, Pak
What do you think,
Nugraha.
maybe?
Can you also mention your affiliation, please, so that the audience understand better?
Yes, I am from Gunung Merbabu National Park,
Right.
under Minister of Environment and Forestry.
Yeah. Pak Nurpana.
Nurpana.
Terima kasih. Thank you for the question, Pak. Actually, we have a lot of students, Pak.
Siap, Pak. Siap dipakai.
Just invite us to analyze the data. Or you may come to our labs. Yeah, we can give a training how to process the data. Yeah, it's not a big matter. Yeah, I think we can contribute on that matter. Ya Pak ya. Terima kasih Pak.
Yes, because the officer of the Minister of Environment and Forestry, so many from the Aceh to the to the Papua. That's very high staff. So many staff, so many institutions, and I think maybe Mr. Prof. Lili make a role model maybe in the institution in the Java. Maybe you can make a role model how to manage with innovation like your presentation maybe.
My pleasure Pak. My pleasure.
Okay, thank you, Mr. Lili.
Thank you Pak.
Can I just follow up with Pak Nurpana a little bit more, because I'm curious with what you just mentioned. So the Merbabu National Park has a program using camera trap. Is that correct?
Yes, yes,
Would you like
we
to-
have done install so many camera trap with systematic grid, 1 kilometer, 1 kilometer, one camera trap.
Mhm. And what are the objectives of that project, if you can share with the audience here?
Oh yeah, next time maybe. Yes, we have monitor every year from the 2019 until now, and we have 116 grid, and we have done more than 50 grid that we have installed camera trap. So many challenge, so many effort from my institution to create that.
That's yeah, that's impressive, and would be good if we hope that you collect the data regularly, download it from the camera, so that I'm sure it's gonna make a good longitudinal analysis by Prof. Lilik. He's excited.
Yes, and the problem is how to analyze that data, not just
yeah.
present and absent only, but
Yes.
also how to occupancy method
Yeah.
and etc.
Yeah, and I'm sure Indonesia has more than enough academics who can solve that problem. So we just need to be better connected with people. Thank you, Pak Nurpana, very enlightening. Any suggestion or comments or questions from the audience, both online and offline? May I ask a question, Prof. Lilik? From my experience with using radio transmitters in orangutans, it's definitely not it's not ideal. Yeah, there are pros and cons. It is cheap. It's quite reliable. It uses radio waves, so we don't need to rely on satellite. However, there are some disadvantages, like short range, and then sometimes, because we put it in primates, we cannot have any parts outside of the body because they will pick on it. So we have to put it under surgery under the skin of the orangutans. My question is, well, actually, we use radio transmitter, we bought it from abroad. So my question is whether there is a technology here in Indonesia who start to produce radio transmitter, because it's, like I said, it's kind of cheap, And I do see the potential of it being used more broadly in wildlife conservation and management in Indonesia. What do you think?
Thank you. Last time I invited by the head of the BKSDA entity when they are releasing komodos from Taman Safari to the Flores Islands. So before releasing, they place the transmitters and the tail, and then but no surgery, just
Glue it
did
on
glue
the
it,
skin.
yes,
Yeah,
yes.
yeah, yeah.
And then for the movement, they are using drones. So the receivers is mounted in the drones, and then drones flies. When they close to the komodos, they
can
download the
capture.
data.
Okay.
So there's I think there's a good step there, just because can more easily to monitor. So we don't have to go to using VHF antennas.
Yeah.
Just fly in the drone and the capture, and it goes back, and we download the data from the drone.
Oh, okay, so the receiver is fitted to the drone.
Yes,
Wow.
transmitters and the commandos.
It's very interesting.
Yeah, this is I think a good
Thank you.
technique. Yeah.
Nice.
Yeah.
Thank you.
And it is developed by company in Indonesia. Yeah. Also, my student also try using for the kura-kura moncong babi in Papua. So they develop the same GPS. Yeah.
Okay.
Yeah. Thank you.
Thank you, Prof.
Using the mobile phone. Yeah.
Oh, okay.
Yeah.
Oh, GPS using mobile phone.
Yeah.
Okay. Right.
And when there are no signal, just put it, and then they got signal, they can receive the data.
Oh.
Yeah.
Very interesting. Thank you very much. Any further comments, question? Can I ask from the audience here who uses any kind of technology that we discussed earlier about monitoring or data analysis or community science, or maybe from the online participants also? Mungkin yang offline dulu, ada yang memakai teknologi yang sore ini kita bahas dengan Prof. Lilik? Are we all students here? BNF mungkin? You would like to share?
Lilik and everyone. I'm Irul from Borneo Nature Foundation. So we are using some kinds of technologies. Yeah, of course, camera traps. The reliable one for terrestrial mammals monitorings. In Sebangau National Park, we are collaborate with Sebangau National Park team and also rangers to understanding the population trend over the years by using camera traps. And then we also use passive acoustic monitoring. Actually, there is a student, PhD student, and postdoc students collaborate with Borneo Nature Foundations, and we conducted annual monitoring by using PAM, passive acoustic monitoring. And then the interesting thing is I think we conduct the same things on the thermal drone, but other divisions, not mine divisions. I just wondering how to differentiate the species by using the Amatron, because when I talk about the characteristic by using PAM, passive acoustic monitoring, each species have different amplitude and also how to call it a graphic in the programs. Two thermal drones have the same approach. I mean, like, do certain species have the certain temperature, and then we can identify them by using the thermal drone.
Yeah, thank you. The questions is if we can identify species or individuals if the difference temperature is about 3 degrees, 3 degree Celsius, difference between background environment and the objects. So somehow the positions or the location is not so perfect. Yeah, somehow they are hiding half in under canopy and half outside canopy, and that's why we are very difficult to somehow to detect the species, even the presence. And the second thing is the resolutions of the thermal sensor. Just this is categorized as military equipments. So in commercial usage, they limited the resolution into some degree of, for example, 20 to 30 centimeters. Yeah. If we need a more clear objects that can be analyzing using the shape or segmented based on shapes, we need very clear shape. Yeah. Yeah. And when we use the thermal drones, they are we tried in the savannah polar rinca is using the fixed drones, fixed wing drones. That's the flight cannot be so close to the object just because they need maneuver certain area for maneuver and landing. Then we cannot get very good resolutions. If you use the quadcopter, you can detect more closures. But the problem is the disturbance of the canopies. Yeah, canopies. Basically, the essential difficulty is to use the thermal sensor. Yeah, we cannot get the clear object, and also somehow we cannot get the sufficient temperature difference between object and the background. Yeah.
Yeah, quickly.
Thank you, thank you. I just thinking that in your presentation, you said that the thermal drone can identify certain species, and I saw the surveys conducted on the daytime, and I just thinking that what if there's a possibility to conduct at the night, and then we get certain species or certain heat over there. Because on your presentation we can identify the species because we understand and know the clear pictures on the daytime. And is it possible to us to conduct the same thing at the night and then can distinguish the species based on the thermal body of species?
Yeah. Yeah, it's ideal, very ideal, but you have difficulties in mosaicing the image. Just because when you do mosaicing, you should have a clear object. So if you taken the data in night, everything the same. So you cannot mosaicing the image. So there are two types of image actually. When you use the, for example, Trinity sensors, the sensors are already calibrated. Means that when you download the data, the digital number of this image is temperature already in the Celsius on Fahrenheit. Yeah. But somehow in some quadcopters, it is not calibrated. So when you download the data, it's still in the digital numbers, means 0 to 255 if you store in 8-bit data. So actually you don't see the temperature, just digital number. That's why ideally we can take night, but difficulties in mosaicing. Yeah, a delay is at, for example, like 5 o'clock in the afternoons. It's, I think, the best, based on our experience. Yeah, thank you. Yeah.
Thank you very much for the very interesting discussion. I'm sure after this we will have more discussion during lunch, dinner tomorrow with Prof. Lilik and his expertise on technology innovation. Let's give a round of applause for our speaker today. If you can still join us here and join the next discussion. For the second speaker this afternoon, we will have online. Pak Taufiq Purna Nugraha, are you with us today? Can we have his video? Yeah.
Yes.
Welcome. So
Thank you very much.
I will just introduce you to the audience. So Dr. Hewan R. Taufiq Purna Nugraha M.Sc is a senior researcher at Research Center for Applied Zoology, National Research and Innovation Agency, or BRIN. His main research focuses on comparative reproduction and endocrinology of vertebrates, zoonosis, image analysis, wildlife forensics, and combating illegal wildlife crime. His goal is to contribute to scientific knowledge, conservation efforts, and the protection of biodiversity through research and applied work in these areas. Recently, he's researching about the study of the reproductive biology of male Bawean deer, or Axis kuhli, as part of the wildlife germ plus conservation, and the conservation of male Javan leopard, Panthera pardus melas, germ plasm. Pak Taufiq, I believe you will be presenting from your screen. Have you
Okay,
had the
I will share my screen.
Share screen?
So hopefully everyone can see the presentations.
Perfect. You have around 20 minutes, and the time is yours.
So thank you very much, Dr. Siska. Ladies and gentlemen, and esteemed college and distinguished guests, I'm really sorry that I am unable to attend the symposia in person, but I'm honored to join you virtually and to discuss on this topic. In my presentation, I will explore innovative technique in reproductive biology that offer hope for conserving endangered species and restoring ecosystem. Indonesia is well known as a mega diversity nations, and we also have like extensive array of fauna species. But nevertheless, a lot of this wildlife species, our asset, is face significant threats, resulting in unfortunate extinction of several species. Data from the IUCN Red List, Indonesia has 4 species that are now extinct. With the 2 most famous one is the Bali tiger and the Javan tiger. And this sobering reality underscores the urgent need for enhanced conservation effort within Indonesia. And not so long ago, we also heard about the death of the last male of African white rhino and the death of the last male of Sumatran rhino in Malaysia. This is really heartbreaking, and to see how this majestic species is threatened to be extinct in our neighboring country. Recent report by the IPBES warns an unprecedented global nature declines and accelerating species extinction. This decline is primarily driven by human activities, and it was approximately 1 million animal and plant species are threatened with extinctions. And also, greenhouse gas emissions doubled since 1980s, and this exacerbating the effect of other drivers. For now, the conservation of biodiversity has mainly relied on the classical conservation approach. Some of the examples is like for species protections, the habitat protections, ex situ conservations, etc. But from what we have learned, the rate of extinction is so fast that we have to accelerate the conservation effort. One of the ways is by accelerating technology that support conservations. Innovations, for example, the application of advanced experimental method, as that has been shown by previous speaker, using AI, using drone, also the citizen science, big data analysis and AI, and also one of the branch innovation is reproductive biology, such as assisted reproductive technology and genome resource banking. In addition to the so-called classical conservation approach, cutting-edge biotechnology can also be applied to maximize and accelerate conservation effort. The application of the new emerging tools can help to expedite this process. One of the examples is assisted reproductive technology and genome resource banking. This is a flow chart by Pierre Comiselli about how the use of these emerging tools can foster the progress for sustainable populations being settled in the wild. And now we came across with this term that I was brought, the reproductive biology technology. This is a technology encompass of a range of techniques that aim to control and manipulating the reproductive process of various species. It offers valuable tools to address challenges in biodiversity conservation, such as preservation of endangered species, the restoration of species to ecosystem, and the enhancement of genetic diversity within populations. This technology are grounded in the principle of assisted reproductive technology, molecular genetic, and conservation biology. One of the things that I would like to introduce here that is already long known is the assisted reproductive technology. This is a medical procedure primarily used to overcome infertility problem in human, but it also has been developed for over 100 years in domestic livestock to increase productivity. And recently, advanced IRT implement recent advance in biotechnology and stem cell related approach, such as cloning, inner cell mass transfer, and stem cell associated technique. This approach are used to generate gametes, ultimately embryo of highly endangered species. So IRT has evolved through generations from artificial inseminations. This is probably something that most people are well known, like what we use to in domestications of like our cattle. This is like the first generations, and we also have embryo transfer, in vitro fertilization, ICSI, sperm sexing, gamete embryo conservation, and for the latest is like the somatic cell nuclear transfer and stem cell, or most people know it as cloning. The main challenge in assisted reproductive technology is for early generations and the species is the collections, preservations, and strategic deposition of gametes. This process is crucial for the conservation and diversity in endangered species. So I will give you some of the example. One of things that we need for application of this assisted reproductive technology is also like the gamete, like the germ plasm itself. Like for the male, we call it semen. Semen is consistent of sperm and seminal plasma. So this is from the male side. And there's like a lot of way to collect this from wildlife species or endangered species. Mostly in the cattle or domestic species, we can use artificial vagina. But for wildlife, which is a bit difficult to restrain, we will need like more invasive procedures such as like ejaculator, catheter, or even like postmortem collection of epididymis if we have like killed species, rectal massage, or we could also like collect it from postcoital. So after the copulations, usually something dripping, and we could like collect the sample from that liquid. This is an example of our work in Taman Safari Prigen and Taman Safari Bogor. We have collaborations for biology reproductions of bawean deer. This is like giving example how we use this process, like from preparing the dart gun to anesthesia the deer, bring into the collecting area, do the electroejaculation. So for people that's not familiar, electroejaculation is like giving impulse, electrical impulse with the probe from the rectal. We try to stimulate some glands, so in order to ejaculation to happen in male. So afterward, we have to again check the sample and to seek the quality of the sample and do the preservation process. This is the example analysis using computer-assisted sperm analysis to check the quality, like the motility of the sperm sample from Rusa bawean. More simple approach like using the artificial vagina also have been tried in our previous work with the banteng bull. But for something that's in the exit to conservations or in BRIN center, it's something that doable, but it will need rigorous training and habituations. So as it looked like simple, but it took like months of training, like 6 months or something. You have to habituate the banteng bull to the keeper, introduce to the different novel objects, so he could feel comfort to see the cute for holding the teaser female. And so we able to use the artificial vagina to collect the semen sample. But recently, technological advancement also has led to availability of sporting devices such as compact computer-assisted sperm analysis tools. This is very handy and mobile. You could like put this in your pocket and do sample analysis in the field without have to bother using manual analysis or even bring a big CASA system for checking semen quality in the field conditions. This is another example of semen collection. We are using the catheter. This is the catheter method. So we put a catheter to go to epididymis of the Panthera pardus. This was conducted in Jogja, Hartam, in Gembira Lokazu. And this is like the result from the sperm collected with these methods. And also from the female side, there's some approach that can be done from the ovum egg collection. So we collect the egg from the female, but this will require more invasive methods such as ovum pickup using laparoscope, ultrasound guide of laparoscope. And also there's like an embryo collection. So if the female has been fertilized or have been used artificial inseminations, when it's developed, the egg is developed, and we could like collect the embryo and to be frozen or to be transferred to another individual. There's also the need of synchronizations of reproductive cycle for female. Also for insemination, we have to know when the female is in heat, and also for collecting the ovarium or collecting the ova. And for the application, right now, the most visible way is using artificial inseminations. This is a very old technique. This is technique to introduce semen fresh or frozen into female reproductive tract. This is in Rusa Timor, deer. But even this is like the most simple and the first early generation of assisted reproduction technology. But this have been proven in the case of black-footed ferret, Mustela nigripes. This is represent the notable success story in application of assisted reproduction technology to reverse extinction. This species declared extinct in 1979, and using quality from male sperm in 1997, artificial insemination is using fresh semen was conducted, and in 2015, insemination using frozen semen because the technology has successfully developed, and currently more than 3,000 individuals has been successfully reintroduced into the wild. There's also a case of the use of in vitro fertilization and embryo transfer, and it has been even done into interspecies, where from eland into gore, this has been tried to transfer the embryo to surrogate mother. This is like the report of the embryo transfer in baboon, and this is the infant resulted from this procedure. So another more advanced technique is somatic cell nuclear transfer, or This is a technique which the nucleus of the somatic cell is inserted to the cytoplasm of an egg cell that had its nucleus removed. So basically, this is like a cloning procedure. I think most of attended here has been heard before about Dolly, the cloning of the sheep in 1996. This is the same procedure. But now this procedure has been evolved, even make it possible to do interspecies somatic cell nuclear transfer. Like for instance, like the somatic cell from cattle, we use from the embryo, and then we use the somatic cell from the endangered species. And now we have collected the sample, the semen sample, and everything. This will be raising another issue, like how a part of this application of collecting insemination, there's a crucial issue how we can store this sample for extended period, say for species recovery. We do the cryopreservation technology using liquid nitrogen, and also now there's this a trend developing genome resource banking. GRB is the concept of conserving the germplasm of wildlife in the form of sperm, embryo, oocyte, tissue, blood, DNA, and other genetic materials through cryopreservations. And this GRB concept has been implemented in several countries such as United States, South Africa, Thailand, and South Korea. And this has been done really long. I mean, a lot of conservation has been collecting samples from wildlife for decades, from decades to save genetic diversity. In development of the GRB, one of the sample that we use is like from in Smithsonian. In Smithsonian, they have 1,000 samples from 70 species. In San Diego Zoo, they have like 1,000 taxon and 10,000 living cell. In Thailand, they have like 20 sperm banks. And currently, there's estimated at least 50 of this biobank in 20 countries. As for Indonesia, this program is still scattered. I heard that the Ministry of Forestry and Environment want to also initiate this program. BRIN is also have this program, but it's also still scattered by research. We also have Sumatran Rhino Sanctuary Program for the Sumatran Rhino, and also ex situ conservation institutions such as Taman Safari Indonesia now is very actively engaged in collecting germplasm from endangered species. This is an example from the San Diego Zoo. They have like an extensive collections. This is also example from the Korean wildlife. Has annually cooperated with the administration to train expert for rescuing and managing wild animals and treating it as national treasure. So these collections, cryopreservations, and things possible. So, but the significant resource required to maintain and utilize biobank sample combined with the large number of treated species requiring conservation intervention globally make it impossible to sample and conserve every species this way. So this is a chart that we propose. So we have to have like the same vision. Like now we have to have like the priority from the checklist database. We have to make scale the priority. We have to have target species. And we also have the literature study, what data has been accessed, what data is not. Biological data is very important from anatomy, physiology, reproduction, nutrition, and genetic. Also, we have to analyze every specimen from every species, chemical composition, sample quality, and also development of cryopreservation technique, because this is can be very different on different species. And hopefully, from all of these result, we could like restore the in situ population of the species. But there's also like a challenge and ethical considerations. While reproductive biology technology offer immense potential for biodiversity conservations, there's also presence challenge and ethical dilemmas. Critics argue that this technique may divert resource from holistic conservation effort because it's very expensive to maintain, and also question about welfare of the clone or artificial breed animal. And there's also potential for unintended consequences such as genetic homogenizations. Like in the sample of the black-footed ferret, it came only from several individual, and now like 3,000 individual. And this can it also be threaten the long-term viability of the species. Another challenge is also very important to optimize species-specific protocol. This is very lab-based, so you have to have like different semen or sperm extender for different species. You probably have different protocol for freezing different species. And this to expand the current knowledge to include all taxa, because mostly based on mammals, but also there's a lot of species that very endangered, like for the amphibian. And also challenge rising from significant diversity in reproductive physiology, behavior, even from the same taxon, like from big cats and also like big dogs, which is very different from the same carnivore taxa. Also, there's like a difficulty in gaining access to the protected or endangered species. We have to acquire permits and everything to collect the sample properly. Also, risk associated with protocol and time-consuming permits, and factors such as quality of human resource. There's a very limited expert right now, and funding for long-term maintenance and impacting this will impacting success. There's also this flagship species paradigm that is like this is a new approach that we start to do. The concept of mostly we are focusing on flagship species to conserving iconic, charismatic, and well-known species, so we can protect the ecosystem and habitat. This mainly as a symbolic ambassador, but to enhance the conservation success, the focus should also shift towards species with sufficient populations, because like with this new cutting-edge method, is very experimental. We need a lot of sample size. We need have to improve the protocol, the methodology. So this focus has also to be shift with species with sufficient population and genetic diversity. This is necessary for a better understanding of population size, genetic diversity, and using assessment scheme like the IUCN Red List. Like we could like project like with animal that will be extinct or facing threat probably like in many years to come, and early intervention of genetic restoration increase the likelihood of successful species recovery. So instead of like focusing our effort of like for the limited endangered species that only have limited species and access, we should also start to shift also to the species that's still like very accessible right now, but probably in the future that will be endangered. This is some activities by our lab since 2010. I haven't really updated the data. We have gone far since this. This is like a sample of what activity that we do. This is from our last project. This is in Rusa Bawean. This is gibbons in Gembira Lokazu, and this is the Panthera pardus, Javan leopard in Cikananga, I think. In Cikananga Rescue Center. And for the future directions, we need to more advancing research in wildlife reproductive biology, because mostly we still like relied on the technology or the data from the domestic animal. So we have to bring bridging the gap between domestic and endangered animal applications, and also ensuring synergy between technology and wildlife conservations. Now, this has been mentioned from the first presentations by the moderator about the cross-institutional collaborations. For example, many universities engage in cryopreservation research even with wildlife species, but afterwards, when the student has finished their PhD or their master degree, they just left the sample in the university and abandoned because it's very expensive to maintain. So you need to buy top up the liquid nitrogen regularly. So mostly it's getting abandoned and frozen cement when liquid nitrogen depletes. So it's like really a waste of this very valuable. Like in South Korea, is considered as the natural, as the country treasure. And also we have to also start prioritizing for more abundant species for cryopreservations. And also something that we need to do now is establishing genome resource banking consortium. So it's not like a single fighter like SRS working by himself, or like the Taman Safari or the Ministry of Forestry and Environment or BRIN. So we have to have sort of like collaborations, interdisciplinary collaborations involving research institute, university, conservation organization, and NGO. So to summarize, reproductive biology technology rooted in assisted reproductive technology offer promising tool for addressing biodiversity conservation challenge, but there's still a lot of things to tackle, and hopefully we could be more collaborative in the future to have the same goal to recover our endangered species. So thank you very much. That's for my presentations. Now back to you, Dr. Siska.
Dr. Taufiq, so his talk is mostly about innovation technology in preserving wildlife, focusing on the current technology in what, reproductive technology? He
Yes,
mentioned a lot.
in reproductive biology, so like the applications of assisted reproductive technology that's already well established in domestic species is also something like new hope to recover for the recover the endangered species that we have right now.
Yeah, it's indeed a very interesting and really what do you say, out of the box solution, for example, in conserving wildlife. So we need to collaborate more with multidisciplinary experts. Even in this case, it's not multidisciplinary. We are talking about the same profession, veterinarians, but sometimes those who are working with wildlife do not really communicate with those who are working in livestock, for example. And we need to bridge that, yeah, Dr. Taufiq. Right. And you also mentioned in your talk about the, I really like this idea, genome resource banking consortium, and that is something that we really need to think about because it's our national treasure, like you said, and it doesn't belong to any institution, either it's academics or government or NGO or anything. It's our national treasure, and it should be really managed together, collaboratively, with as many parties as possible. Thank you very much. Do we have any question or comments about reproductive technology for wildlife conservation, genome banking? I would like to ask our other expert panels here, Prof. Lilik or Prof. Hong, do you have any comments about genome banking? Maybe Prof. Hong, from your background in Korea, how is it going?
Well, actually, the subject is very far from my field, so talk was very interested, but I have no questions.
Okay. Thank you. Anyone? Maybe from the online participants? Oh, silakan, Prof. Lilik.
Terima kasih. Pak, ada one question is about is it possible to, for example, to make for example, if we want to what you call menghidupkan kembali.
Oh. Revive, to revive extinct species.
Yeah, already extinct species, using, for example, birds, but we use chicken, for example. Is that possible?
Yeah, I think that approach that we try, I mean, a lot of researchers try to accomplish with this called interspecific somatic cell nuclear transfer. if you have heard before, there's like an effort by some researcher to revive woolly mammoth. So they got this really nice sample, like frozen woolly mammoth, with still like good intact tissue, and they were expecting like, or they try to clone like from this somatic cell to take the DNA information and then probably put it on, for example, on the elephants, nowadays elephants, and then to see whether it could resulting in born a new woolly mammoth. So that is something that some researcher group is trying to achieve right now. So for now, it's still like highly experimental, just like Dolly. If you know, like it's the longevity is not long. We still a lot of things to work with, but maybe in 10 or 20 years, or probably like next year's, some breakthrough has happened, and then in this area, and then people could start try to recover species, extinct species from the sample that we have now, like in the museum and everything, and to have to do this procedure like cloning.
That the extinction species recovery could be debatable. So, what do you think about the reintroduction of the extinct species?
Yeah, it's something also like in the realm of this ethical
Yeah. Yes.
area
Yeah.
about this issue. For me as a researcher, that will be very interesting subject, of course, because I'm working in this area. So for me, as a researcher, this is really interesting. But regarding the ethics, it's something that has to be discussed, not just by the researcher, but also from the conservationists and everything, and to see is there will be any impact of restoring this extinct species.
I completely
For
agree with you.
scientists,
Yeah.
for scientists, this is very interesting. But if we see, like for example, if we see like from the science fiction, like for Jurassic Park,
Yeah.
this is like one like an example, like reviving extinct species. I mean, the technology, the cloning, everything is something that's possible, doable, but we are not there yet. But I don't know in the future. So yeah. So ethic-wise, this is something that has to be discussed with multidiscipline and with multi-approach. So not just like from our side, from the biology reproductive researcher, it is something that really interesting,
Yeah.
and we are eager to see if this are possible to be done.
Prof. Hong, you have any further comments?
Essay also one of the subject to discuss, but I think also the ecologically
Yeah.
extinct species recovery could be
Yeah.
positive or negative. We
Yes.
can think
Yeah.
about it.
I guess those species who extinct not because of human causes, they're extinct because of reason, because the nature doesn't fit them anymore. If we want to revive like woolly mammoth, who wants to work with woolly mammoth here? What are you going to do with them?
That is like always like two side of story. But I think something that's also need to be emphasized with this approach. I mean, if we just like talking with the only with the living species right now, is the collaborations. This is something that's we see is running a bit slowly here. I mean, if I talk with my colleague like in Thailand, they have this very good collaborations. For example, when the zoo is going to do anesthesia or do sedations to their animals, they will call the university, so it will come and also help them to collect the gamete, to collect the sperm, so they don't have to do like do sedations or do anesthesia to some animal just to collect the sample, just like how we do it right now. So it's kind of interesting if we have this kind of collaboration also in Indonesia to have, I mean, like, for example, like in orangutan rescue center, every time new orangutan coming, we could like, some people could also collect like the semen sample or something that we could preserve, and as this is just as a backup in case some catastrophic happens with the nature population. We still have a lot of sample with very diverse genetic, so it's not just like from one or two individual, but we have like many individual that could represent a healthy genetic population compositions.
Yeah, that's a very good suggestion, and I think all of us here are agreeing with your suggestion that universities should be more practical, if I say, and be involved in the real conservation actions by learning, by applying technology, doing research, and those kind of things. Anything else that anyone has a bright idea or comments or questions to be raised in this discussion? Prof. Lilik?
Yeah, this is a big issue about the Jawa rhino, Pak. It's a scary story, yeah, if the species would be extinct. So is there any activity to collect sperms from this species?
Now we're talking about rhino, the most difficult wildlife to reproduce, I think. Any thoughts, Pak Taufiq? Sorry, you are still muted, I think?
I think like this 7 and 8 of September, there will be a meeting about this about this assisted reproduction technology in Javan rhino, if I'm not mistaken. I was our BRIN was invited to the meeting on 7 and 8. So I don't know if that something has been done right now. I haven't heard, but I think it will be try with the Javan rhino. But I know with the Sumatran rhino, they already have this program in SRS, in Sumatra Rhino Sanctuary.
There might be plans for artificial insemination in Javan rhino.
So the thing that I want also to emphasize, Doxiska, is about we should act now while we still have like plenty of individual, instead of like waiting for in case like the one that happened in Borneo in Malaysia, where they have like the last one male. It's they have the quality of the sperm is bad, it's difficult to collect and everything. So it's very, it will be reduce your chance by like 80% or 90% if you only have like several individual left, like the one happened in like in Africa when it's already extinct. So yeah, I think some dramatic effort has to be done and not wait until we only have like several individual left in the wild.
It all comes down to prevention. Make sure that we still have our ecosystem, our wildlife intact, and it's much easier, much cheaper, and much better for us humanity rather than, relying on technology and trying to revive
extinct species. Yes, of course.
Species. Thank you very much, Dr. Taufiq, and all the
You're
audience
very
for
welcome.
the lively discussion. Let's give a round of applause to everyone here. Okay. We're getting there. We'll have our last but not least speaker of this afternoon, and I'm sure the last speaker will be as interesting as the previous ones. May I present you Professor Sungwon Hong, He is an assistant professor at Department Horse and Wild Animal Science, Kyungpook National University. His research interest is in wildlife ecology and management, invasive species management by spatial analysis, food web structure analysis by stable isotope and DNA barcoding, endangered species management by spatial and temporal analysis, and population dynamic by genetics and modeling. Currently, he participated in projects such as African swine fever in wild boar in South Korea, ecological survey of national park, and K-Water open innovative R&D. Professor Hong, you have about 20 minutes to share your talk, and then we'll continue with discussion. Time is yours.
Thank you for introduction. Hello, my name is Sungwon. And actually, throughout my postgraduate study was to investigate how Eurasia otter recovered, even though South Korea has been rapidly urbanized. So one of my aim was to discover the contribution of environmental factors. So this topic is quite far from the aim of this symposium, but it could be interested for the developing country, for example, Indonesia. So my topic is about the otter recovery in South Korea, the contribution of environmental improvement and conservation policies. So we conducted 5 national surveys throughout 1993 and 2019, and then the result showed that otters has been rapidly recovered throughout the country. But the important thing is that each survey has different survey efforts, so that there are some bias between the surveys. But even though the survey could be biased, but currently otters are observed throughout the country, even though in metropolitan city such as Seoul, Daegu, and Busan. And so, for example, Seoul largest city, minimum individual is 61. As you can see, the red color is represented the urbanized area. So the South Korea rapidly urbanized. So my research question was, how have the otters been recovered? So my hypothesis was that the water quality improvement affected the otter recovery. So as national incomes from industrial development has been rapidly increased, for example, compared to the other country, South Korea, urbanization rate has been rapidly increased. And then, according to the industrial development, the national income has been improved. Then increased national income allowed the government to invest the money to improve the water quality. So, for example, we invested $410 million to improve the Gumon rivers. So BOD was 191.2 in 1983, but nowadays only 3.6. So high BOD means high polluted areas. So we invested the high budget of the national income, and then the water quality has been improved. And then second hypothesis is that the enhanced protection law affected otter recovery. So in 2005, the government classified the endangered species into first and second grade to enhance the protect the critically endangered species. And then the legislation included the provision for the punishment of hunting, trading, and consuming otter product. As I briefly introduced the South Korea is a very small country, but and then the temperature is the climate is temperated, but the human population is very high compared to the other country. And then as you look at the land cover map, the eastern part of the area covered by the mountains, almost 62% of the area. And then as you look at the red color is the urban area. So urban area is located in the middle and lower middle rivers. So we have 5 large rivers: Han, Geum, Yeong, Nakdong, and Seomjing rivers. And the most high altitude area with the high altitude located in the eastern part and the middle of lower part of the South Korea. And then we manage the water quality based on the 111 river sub-basins. So we reviewed the 4 national water surveys spanning 17 years, and then the water distribution map were overlapped with those of the 111 river sub-basins to define the water detection and non-detection history in the river sub-basins. And then also I conducted the field survey at the 2016 and 2019. And then because for only one person to investigate the water survey could not be enough for 1 year. So I divided 2 years. So 2016 I conducted the Nakdong, Seomjing, Yeongsang rivers, lower part of the river, South Korea, and then I conducted Han and Geum river in 2019. So at the site, I walked 600 meters along the riverway and then calculated the density of fishes, which is the normal monitoring method of the IUCN water specialist group. And then I collected environmental variables: forest density, percentage of urban forest water protected areas, which could affect the water distribution. And also I collected water quality data, which is my hypothesis. So I collected the water quality data, especially BOD, in each river sub-basins. And the environmental data were extracted for each river sub-basin in each survey years, so 2000, 2005, 2010, and 2015. And then I conducted the single species, multi-species occupancy model. So this model could dealing with the detection probability to reduce the bias from different survey method. And then this method also current presence could be affected extinction and colonization probabilities. And as you look at the more precisely, this model has 2 sub-models, which are the initial water occupancy model in the first survey period, and the second is the water colonization and extinction probability with detection during the later 3 survey period. So this is the details of the modeling. So I conducted the Bayesian approach using the JAX package, and then I applied the multi-model inferences based on the lowest Watanabe Akaike information criterion. And then I conducted some goodness of fitness using the Bayesian p-values. So the result show that during the initial period, water distributions in river sub-basins, waters was distributed in the area with high forest densities. But interestingly, colonization and extinction probabilities are highly dependent on the water quality. So this means that initial period, waters are survived in the forest high police density, which mean that the high police density could be could represent the area with the remote far countryside avoid from the human disturbance. So at that time, otters are survived in the area, and then nowadays only water quality is water recovery is dependent on the water quality. So presence probability of otters has been increased during the period as you look at this plot. So this plot show that my proved my hypothesis is otters are recovered, but interestingly, as mapping the presence probabilities, some areas are still vulnerable. So we should improve these areas. And then as you look at the relationships between BOD and otter colonization and extinction probabilities, very interestingly for me is that between the second period and third period, the relationships are changed. For example, for extinction probabilities, the coefficient has been relatively remarkably increased. Otherwise, for colonization probability has been changed from negative to positive. So as we can explain that the otters could be colonized in the unpolluted area, but this means that nowadays otters recolonize the area with the polluted area, because BOD, high BOD means the high polluted areas. So that a positive mean is that otters recovered in the polluted areas. So it could be not make sense. So as we look at the mapping the extinction and colonization probabilities during the first and second period, extinction and colonization probabilities, it is still interesting that between second and third period, the relationship has been changed, so that extinction probabilities during the first and second period, the probabilities were homogeneously moderately high, but since second period, the probability became increased in the west area in opposite with the pattern of eastern areas. Otherwise, for colonization probabilities during the first and second period, the probability was high in the eastern area, but then in the between second and third period, homogeneously quite low throughout the country, and then opposite patterns showed in the between the second and fourth survey period. So why? So we should interpret the result, and then as you look at the protection law, the area protection law has been enhanced was the 2005, which is the between second and third period. So that before the enhanced law established, the author's extinction probability quite homogeneously high, because hunting is allowed throughout the country. But then nowadays, only otter extinction probabilities are relying on the environmental variables, because enhanced the law. Otherwise, for looking at the colonization probabilities, after protection law has been enhanced, the authors try to do colonize throughout the country. So after the right after the protection law enhanced, the colonization probability is homogeneously high, and then nowadays, because the area with the high suitable areas were already colonized. So authors tried to in the area with the polluted areas. So when the auto population has been saturated in the high suitability areas, and then the colonization probability were high in the area with polluted areas. So we could say the buffer effect in ecological term. So we concluded, according to the DG result, that because the South Korean government wanted to try to go down the grade of the endangered species for otters, but I could say no, because our otters are very highly dependent on the protection law. And then also I suggested where to improve the habitat could be the western part of the South Korea, and then what to improve is the water quality based on the BOD. So thank you very much. But I became professor. The 3 years already passed since I became the professor, but I just continue to study about the water recovery. So very honored to present my research for you. Yes, because we are experienced the developed countries. Thank you.
Thank you very much, Professor Hong. Let's give a round of applause for his interesting talk. So he shared his research about otter, which is, may I say, urban wildlife. I've heard a lot about otters also in Singapore. That's another story, but they have recovered the population of otter in Singapore. Basically, to summarize Professor Hong's presentation, he investigated 2 factors that might affect otter population, which is environment factor or the water quality, and human factor or law enforcement. And I find it very interesting that only after the law enforcement factor is stabilized, then we can see the effect of environmental factor on the species. So it kind of show us a very good example that human is indeed a very crucial factor in wildlife conservation in general, and this example about otter conservation is a very good one. Do you have any comments or questions from online and offline participants? So may I ask you, Prof. Hong, because you mentioned that before the law enforcement was put into effect, the population of otter is very low. Why is that? Why are people hunting otter? Do they kill them for production or for competition for fish or whatever?
Actually, in 1960 we was very poor, so when I talk with the older persons, they hunted for food, but in 1960s, my study year is just to 1990s and 2000s. So what I'm saying is that the conflict between the human with the otters are
For food. Oh.
For fishery.
Right. Yeah.
Yes,
Okay.
fishery. So nowadays the fishermen also argue to decrease the grade of the protection, because the otters are now protected by the 2 governments, so Ministry of Environment and the Ministry of the resources.
Right.
Resources mean the natural and the historic.
Right.
So
Right.
the
Okay.
so very enhanced protection law allow the otters recover
Yeah, I think it's about the same issue with us here, yeah, that in Indonesia, otters are also, in some areas, in urban areas, they are considered as pests because they eat fish in fisheries. At least in Java, that's what I know. And yeah, it's really a fine balance between protecting the wildlife and keeping the human happy.
The important thing is that I focus on the otters, but then nowadays South Korean some mammals have been recovered nowadays. So many mammals wildlife are recovered. So I would like this research to enlarge into insight how mammals could be relying on the protection law or human protection like that.
Is it river mammals or even terrestrial mammals that you're talking about?
Even other mammals as well.
So land mammals also you
Yes,
see kind of increase in
yeah,
the,
yes,
oh, wow.
land mammals. So the terrestrial mammals. So and then one of the important thing is if government increase the national income increase, government try to improve the environment as well. So with South Korea also forest density has been increased as well. So mountain environment has been much improved since Korean War. So I also would like to talk about the human activity change due to the high national income. So,
Right. Okay. So you're saying that first up, because of the otter population are recovering, you see that there's improvement in the ecosystem of the wildlife, and also you mentioned that after the human economy improve, you see also improvement in the interest of conserving wildlife.
Yes,
Wow.
human economy is very important. For example, we try to reintroduce some critical endangered species, but we only focus on the survived species, not extinct species. So, so I would like to express that to protect the very critical endangered species is also important.
Yeah, yeah.
Because after the extinction, and then people with the high national incomes, but people do not want to restore the extinct
Okay.
species, more animals. For example, tiger and leopard in South Korea.
Oh, okay, okay, okay. So, yeah, I guess Professor Wong wants to appreciate that us here in Indonesia, we really should conserve wildlife, especially because we are mega biodiversity in the world. And based on his perspective, even after a country is developed, they still want to conserve wildlife because they are serving the ecosystem, and it's very crucial for humanity. Any comments, Prof. Lilik? Maybe you have some insight to be shared with us?
Thank you for your nice presentations. Just wanting to know what is the predator of the otter in Korea, and then when you develop the model, how you decide the variables to be included in the model, for example, contections, forest density, and so on. So have you any prior analysis before to get the?
Yes, I actually several study was conducted to know how otter recovered. So, and as you look at the field monitoring system, my voice. So I conducted, since 2014, I conducted the monitoring for 10 years in Nakdong River Basin, one quarter of the South Korea areas, and then I defined some environmental factors, but I also referred some other literatures. The first question was, what is the there are no predators in South Korea. No predator. I think
In the past, like
naturally maybe tiger, leopard, but crocodile we don't have. But and then the no, snake is smaller. And then we restore the Asiatic black bear nowadays, but this is the restoring species. So it's set for the restoring species. Otter is the most biggest mammals in South Korea. Most biggest.
the biggest.
Yeah, totally.
But if you say the top predator is human, yeah?
Yeah,
Now
very-
it is human. Sorry. Thank you very much. So earlier we discussed about what is the predator of otter in Korea, because they are native to Korea, so there should be, food of chain. There's got to be someone who eats the otter or keep the population in control. But apparently, it's very hard to even historically. It's not really clear what is the higher predator of otter. And the Professor Hong said that otter is like the biggest mammal in Korea. Currently, is the biggest mammal. Any thoughts, comments before we come to the end of this session? If not, then I would like to again ask the audience to give a big round of applause to our three distinguished speakers for the insightful discussion. Thank you very much for your active participations, and I will return to the MC of the day. Thank you. Thank you very much. Very interesting.
We graded our thanks to our chairperson here, Dr. Francisca, and all distinguished speakers for your invaluable insight today. And thank you for all being here, ladies and gentlemen. And let us commence this symposium with a sense of purpose and a commitment to making a positive impact on the future of wildlife biodiversity conservation. And we are so happy since you are still here to listen the last session of today.
Yes, we hope we can meet everyone again tomorrow on the last day of WBC of 2023. For tomorrow's agenda, we have a workshop, closing ceremony, and a special lecture from Professor Richard Collett from the Center for Integrative Conservation at the Shizhuangbana Tropical Botanical Garden, Yunnan, China. Before we close this session, we would like to remind everyone to please join us in the front of the room to take for a photo session. So after this, please everyone could join us in the front. Okay. Without further ado, we'd like to close this session today. Good afternoon, everyone, and we hope to see you again tomorrow. Thank you.
Thank you. Please, ladies and gentlemen, come and join us here in front of the screen to take a picture together as our documentation of second day of this symposia.